Dear students, so in the last episode, we were talking about resting membrane potential. We were talking about the mechanisms which are responsible for resting membrane potential to happen, to occur, and to maintain itself. So we talked about three main mechanisms: electrogenic ion pump, sodium potassium pump, the Dohn equilibrium, and diffusion potential. We talked about the first two, electrogenic ion pump and Dohn equilibrium. So just for a brief review, electrogenic ion pump or the sodium potassium pump is responsible for moving three sodium ions outside the cell and bringing two potassium ions inside the cell. So three positive charges are going out, two positive charges are coming back. Balance is -1 for one ATP molecule. So what happened that it is actually creating more negativity inside, more positivity outside. Next is Dohn equilibrium. We talked about that if a membrane is permeable for a few molecules, then molecules will move from one place to another place, just like from inside the cell to outside the cell towards extracellular space and towards intracellular space, ions will move and until a balance is maintained. Equilibrium is reached, and that is called Dohn equilibrium. But in the cell, we said that this equilibrium is actually shifted towards more negative ions and molecules remaining inside the cell, like proteins, DNA, RNAs, many negatively charged molecules. So ultimately, more negativity is there inside than outside. So most of the negatively charged molecules cannot go out and they stay inside. That's why there is more negativity. Third thing that is responsible for generation of this resting membrane potential is called diffusion potential. We know that diffusion is a process of the movement of the ions molecules from their area of higher concentration to the lower concentration. Now there are three important aspects in electrical contribution to the membrane potential. One is called ion mobility. That is how mobile are the ions. Ionic concentration differences. What are the generalized concentration differences inside the membrane and outside the membrane? That is intracellularly and extracellularly. And ionic permeability. How permeable the membrane is for the ions, movement of ions which are there. Let's talk about them one by one. So ion mobility means that different ions have varied mobility due to different sizes and charge. So one thing, size, the other thing, charge, and what we call charge density. Now what's charge density? What happened that when ions are there inside, size we understand. Size means how big they are, how small they are. Of course, larger ions generally should have lesser mobility, smaller should have more. But there is a difference in the cell, inside the cell. What happens that you know when when any ion we are talking about, it is having a charge like +1, +2, +3, and this +1, +2, +3 refers to the valency shell electrons basically. Um, I think you have to recall the valencies and so on. But +1 means that there is +1 charge around that, and one electron is moving here and there around it. Then +2 means that there are two, and so on. Now what happened that when these ions are inside the cell, actually they are having a hydration layer. So when an ion is inside, it is actually having a hydration layer, that is water molecules associated with it around it, right? So like this, water molecules are surrounding it. It depends upon the charge density, that how many molecules of water will surround this. Like if some molecule is having, if both molecules are having +1 charge, but one is smaller in size, the other is larger in size, like this one, then what will ultimately happen that the one which is smaller in size, in this one actually the charge density will be lesser, because the the charge means that electrons are moving around it and making a cloud, what will happen? That more larger the size of the ion, lesser will be the density of those clouds which are made by movement of electron. So it means that when electron is moving around this one, it's taking actually smaller circles. When it is moving around this one, it's actually going for larger circles. See, moving all around this. Here it is moving all around this, so quicker. It means that this smaller one will attract more water molecules than this larger one. And this large one, in comparison, will have less charge density and hence attract lesser water molecules. So what happens ultimately? It's like this, that more the charge density, more water molecules they attract, and ultimately the size of the ion, that is basically we call it hydrated ion size. That is the ion size which is due to hydration layer of smaller molecule will be actually larger. So if we compare these both, then what will happen ultimately, that due to the hydration layer, the size of the smaller ion will actually be bigger than the larger ion, because here there is smaller hydration layer, here there is a large hydration layer. So size of the small ion is actually more if it is hydrated. So we can also say that hydrated ion size will be bigger, larger for the smaller ion. It means that small the ion, large the hydrated size. Small the ion, large, we should say more clearly, smaller the hydrated size. Now it affects a lot. See, for example, if there are some ions which are there inside, and when they come inside they are hydrated. This one is hydrated, this one is hydrated. Now its size is smaller, its size is larger. This can go out, this cannot go out. So membrane become, the semi-permeability of membrane hence work in this way, that the different ions have varied mobility. Now this one is inside the cell, despite its larger size, more mobile. This one is more mobile because it is having a lesser hydration layer. Due to hydration layer, its size is smaller now and its size is larger now. So this is how varying hydration layers are actually causing affecting the mobility of ions, and hence the potential difference that is generated. Second is ionic concentration difference. Inside the cell has basically lower sodium and chloride concentration and higher potassium concentration. There are similar concentration differences of other ions across membrane. Now we see sodium is having +1 charge, chloride is having -1 charge, potassium have +1. So you can see that these are, and there are more ions. Just like this, somehow more negativity is created inside the cell and more positivity outside the cell due to these concentration differences in the ion across the membrane, say inside and outside. Third thing is ion permeability. That is basically based upon the previous two. There is a selective permeability of the selective permeability of membrane to different ions due to hydration layer, due to their size, due to the charge density, due to their +1, +2 different charges. The ion channels impart their characters. Now, why this difference happen? Because we know that the cell is covered by a membrane that is bilipid membrane, and these ions cannot directly move from one place to another place like outside and inside the cell. How these ions move? Always they need ion channels. So protein channels, the ion channels are actually very important. This characteristic, that is permeability of the ions, is completely based upon the presence of ion channels. How many kinds of ion channels are present? Now the neurons have specific number of ion channels of specific type. Like for sodium, there are different types of ion, there are different numbers of ion channels. For potassium, there are more. For calcium, there are others. For chloride, there are others, and there are more. So there are different types of ion channels. Now, a few of these ion channels are leak channels. Leak channels mean that these ions can move freely. These are always open, and without the use of energy, ions can move across. But few are based upon utilization of energy, and they go mostly against the concentration difference. We will talk about them later when we will talk about action potential. So basically, due to ion channels, there are selective permeability to various ions, and that is responsible for this difference, potential difference of ions, membrane basically, inside and outside. These channels work variedly for ions permeability at different times. Now, see, for example, if sometimes the channels are open, sometimes the channels are closed, and then when the ions are outside, they are having different hydration layer. When they are inside, they are having different hydration layers. This is also responsible for the varied work of the ions, ion permeabilities of these channels. If channel is closed, ions cannot go out or cannot come in, and like that. Now, channel action is understood by their channel blocking agent. How we got to know that these channels work in this way? See, if everything is going all right, we never know that how they are working. But if sometime there is abnormality happen, we get to find out that why this happened, and ultimately we understand the actual mechanism which is normally happening, and now this is stopped like that. So how we understood? There is a toxin. There are few toxins like tetrodotoxin from the pufferfish. It basically blocks the sodium channels. What happened that tetrodotoxin have structures similar to the to the ions, which have to go and attach at a particular place, and it attaches at that place, and so sodium cannot enter inside. So it actually can block the action potentials, resulting in, of course, very serious serious effect upon the person or the animal or human being. Another one is called saxitoxin. It also blocks the sodium channels in dinoflagellates, which are passed to shellfish. So when we got to know that if some pufferfish or some other organism, it injects a poison, we call them poisons. What toxin? When we get to know that what these toxins are actually doing, we get to know that how they can block it, and then see, oh, this is how this action was taking place. So now scientists can actually use the tetrodotoxin and block the specific sodium channels and see what's the effect upon the animal, or what's the effect upon the body of animals of that potential difference. So if sodium channels are blocked, see what happens. This is how we got to know how these channels act, because we can block them, say, and then we follow up that what is the cascade of actions which are taking place and which actions are not taking place. We get to know that these sodium channels were responsible for these actions because we have stopped them, and now they are not happening, and we get to know. So this is just like that. We use these toxins basically for research, to find out, to block some specific channels, and then see what's the effect upon the overall body, which actions are taking place, which are not taking place. So we get to know that these actions are due to opening of sodium channels. Now they are blocked, so this action is not happening. Like for example, action potential is not producing in a particular part, and we know that this is due to so action potential was happening due to sodium channels. It is blocked. Now action potential is no more. So just like that, we use these things to understand the channel action again and again. So channel blocking agents are used for research upon the action of the channels, and this is very important in all kinds of neurological, for example, diseases or any kinds of issues. So summarize, resting membrane potential is generated due to three mechanisms, which are called which are electrogenic ion pump, domain equilibrium, diffusion potential, and all of these collectively produce and create, we can say, generate and also maintain the resting membrane potential. And if it is there, then action potential is produced. Next time we will talk about action potential. Thank you. you have any question, you can post these.